Good morning, everyone. Please confirm if I'm audible and if my screen is visible to all of you. All right. So today is day 11 of this concept review series. So accordingly, we are going to discuss a very, very important topic from Unit 11. Unit 11, as you all know, is the evolution and behavior. Now, which topic we are going to discuss today is population genetics. A very important topic from your exam point of view, because from the entire chapter you get a lot of questions. So you have to be very thoroughly prepared in order to face these questions. I must say in the start of this discussion itself that population genetics, no, you don't have many formulas here, though it is genetics, so obviously it is numerical based, but not many formulas, only few formulas. But the trick is to understand the questions, to understand the statements, what they are asking. That is the trick. If you go wrong in reading the statement, you will go wrong in the in answering. Otherwise, it is really easy. So, shall we start? We will, in the course, discuss concept and also go through some questions and also try to, use some means of recollecting. So, are you all ready? Shall we start? If you're all ready, let's type BIO. Bring it on. Let's see how many of you are ready. Okay. Okay. Right. So if in the process you have any doubt, any question, you can definitely ask me. Feel free to ask me. Okay. Now, so we are going to start discussing about gene pool and gene variability, because this is what we need to understand first when we are talking about population genetics. After that, we will go to the very standard Hardy-Weinberg equilibrium, and we're going to talk about a very important concept, which is genetic drift. And then we will wrap up by discussing the deviation from the Hardy-Weinberg equilibrium. These are the four things which actually sum up the entire chapter. So to begin with, genetic variation. A word or a thing which is very, very important. Why is it important? Because the more variation you have in the population, the better suited you can be to any kind of changing environment. So, what is genetic variation? It is a measure of the genetic differences that exist within the population. So, it is a measure of the genetic differences. Now, these differences, they can lie in the DNA segments or in the genes between different individuals. So it can be in the DNA segments or it can be in different forms of the genes, what we call as the alleles. So more variation at the genetic level, more number of alleles for a particular trait, these will all enhance or increase the genetic variation in the population. And as you can see in the picture given here, the shells of Donax, you can see that say there is so much of variation here, so much of variation. Okay, so what happens whenever there is a change in the environment? Okay, and one allele is preferred over the other alleles, or if I say in the terms of genes, one particular gene is preferred over the other particular genes. If you don't have the other options, no, then natural selection will never get a chance to select, because natural selection only works in a multiple choice question kind of a pattern. So you have to give them multiple choices. Only then natural selection will pick out the best suitable answer. Okay, so genetic variation gives that multiple choice to natural selection. So your survivability, your chances of survivability increases. So all these differences together comes under gene pool. So a gene pool can be defined as all the genes and the different alleles present in an interbreeding population. Interbreeding population, or what we can say, is species. So all the genes, all the different alleles which are present in a particular species population, that constitute the gene pool of that species. Again, if you look at the diagram here, can you see the different variation that is present? You have X11 homozygous X1, you have X2 X3, you have X1 X2. Basically, if you look at this variation here, you will see there are three alleles, only three alleles. X1 is there, X2 is there, and X3. But with the three alleles, you could form so many combinations. Just imagine what will be the set of combinations if you have more than three alleles, four alleles, five alleles for a particular gene. And yes, it is possible. It's not mandatory that a gene is going to have only two alleles. You can have three alleles. A very common example is that of the ABO blood group, right? Apart from that, genes can also have several copies, multiple copies. All will come under your multiple alleles concept. Now, a gene pool, if you see, like this is the gene pool of the dog species. So you have different breeds of dogs. But if you take all the genetic combinations, genetic makeup from each of the breed, then can you imagine what a large gene pool you have, right? All the alleles which are being deposited in this pool. That means the collection of alleles, so many variation. That is why you can create so many breeds, so many variations. Now, when you are talking about a gene pool, you have two attributes to the gene pools. One is the genotypic frequency. Okay, in that population, let me now talk in the terms of population, because ultimately it is the genetic combinations present in that population which is getting into your gene pools, right? So genotypic frequency, this is one attribute which is very important for us to understand, for us to maintain, because that will tell us what is the variation. Okay, this is one way of measuring the variation. So how to calculate my genotypic frequency? Well, it is nothing but the number of individuals of a particular genotype. Okay, say the genotype AA, homozygous A. Number of individuals who are homozygous A divided by the total population. Okay, this is your genotypic frequency of AA. Similarly, if you want to measure the genotypic frequency of heterozygous, it is number of heterozygous individuals divided by your total population, which is given as your N. The frequency of your homozygous recessive is equals to the number of homozygous individuals divided by the total population, which is N. Yes, exactly. Okay, your name is appearing as don't be biased. I don't know what is your name. But yes, gene pool, as I said, it is associated with the population. Because, see, in the population of a particular species, okay, three things are connected here. Let me clarify this point. We are talking about population of a particular species. Be very specific. All right? So, population of a particular species. All the genetic combinations that I have here, all the possible set of genes that I have here in this entire population, they are comprising my gene pool. Understood? Yes, Kirtana, good morning. Right. So, this is my gene pool. Okay. So, in my gene pool, obviously, I can measure the variation by calculating my genotypic frequency, or I can measure it by calculating my allelic frequency. I'm coming to that. I hope I could make myself clear. Okay. Now coming to your, yes, so if you listen carefully, I have already mentioned this, okay? Population species, I have already mentioned that. Good. Now coming to allele frequency. Allele frequency is talking about how many alleles of particular kind you have divided by the total. Like if I want to calculate the frequency of capital A, okay, this is one allele. So this will be the number of individuals who are having this capital A divided by the total number of locus or total individuals, okay? Total number of locus comprising of this capital A. Clear? So how many, total number of locus of the alleles, I will say. Okay, the total alleles for this gene. That is your allelic frequency. So now, depending upon what data you are getting, what data you have in hand, you have two ways of measuring allele frequency. Two ways are there. The ways are completely dependent upon the data type. Fine? Now, first and foremost, if you have the number of individuals of a particular genotype, okay, of a genotype I'm writing, of a particular genotype, if this data is given to you, then how you can calculate allele frequency is by this formula, which is telling you the frequency of A is equals to 2 into the number of individuals who are homozygous A plus the number of individuals who are heterozygous divided by 2 into the total population. Here N is again the total population. All right. Now see. Similarly, if you are calculating for the frequency of of recessive A, then it is equals to 2 into the total number of individuals who are homozygous A plus N into the number of heterozygous people. Okay? The number of heterozygous people divided by 2 into the entire population. Now, why are we multiplying 2 here and not here? This is a very important concept that you need to understand. See, any formula is based on a concept. If you understand the concept, it will be very easy to memorize the formula. You will not find it difficult to memorize the formula at all. Even in the examination hall, in midst of all that tension and nervousness, you will be able to recollect the formula if you understand the formula. What are we doing? We are trying to measure the number of the locus comprising of capital A, or I can say we are trying to calculate the capital A locus, right? So therefore, in a person who is homozygous dominant, you have two locus which are having capital A. Now, when you're counting number of individuals, that means what? You're just counting one individual here, but that one individual has got two locus comprising of the same allele. So, in order to, you know, calculate these two locus, what we do, we take it as a 2 into the number of individuals. Clear? Now, if you look at the heterozygous is just having one copy of this dominant allele, isn't it? So if I just simply count the number of individuals who are heterozygous, it is equivalent of counting the number of locus comprising of this capital A. Understood? Vishal, I'm coming to it. Just 1 minute. Hold on. All right. Now divided by 2 into n. Now why 2 into n? Again, because we are all diploid, we have two locus for each allele. Okay, for each gene, we have two locus. We are all diploids. So that is why it is 2n. Now, this is one formula. If your data is not the number of individuals, but it is given as the frequency, okay, the frequency of a particular genotype, if this is given to you, any genotype, then you go for the next formula. If this is the data, so frequency of FA will be calculated as frequency of homozygotes plus half of frequency of heterozygous. So Vishal, what you have written is absolutely right. It just depends upon what data you have received. All right. So there are two ways of calculating allele frequency. Remember that. Don't get confused. And let me again tell you, you get questions from these parts, okay? These calculations. So here I have a sample question for you. Would you like to try? Well, it is 2n because we are all diploids. We have two locus for each gene. Okay? A gene can have two alleles or three alleles or four alleles, whatever, but we just have two locus for each gene. We are diploids, right? That's why it is 2 into n. Each individuals are having two locus. I hope you have understood. Yeah, okay. So everyone, try out this question. What they have given here is, in a population at Hardy-Weinberg equilibrium, the genotypic frequencies are F of 1, 1 given as 0.59, F of heterozygous, that is 1, 2 is given as 0.16, F of 2, 2 is given as 0.25. So what are the frequencies of the two alleles at this locus? Okay, Vishal has given me the answer. Kirthana has given me the answer. So you have to calculate for both A1 and A2. All right. Have you all done this? Shall I start discussing? Okay, I'm again getting mixed answers. Yes, Kirtana, I've got your answer. I'm waiting for others. Some of you have given me option 3. Some of you are giving me option 2. You know, you can, if you do one, if you find out one, you can get the other one also. You can come to the answer. They're asking for the 2 alleles. If you find A1, then also you will be able to give the correct answer. Okay. Yes. So, which formula to use here? The data given is the genotypic frequency, right? So, I will use formula 2, which says that the frequency of A1 is equals to the frequency of homozygous A1 plus half into the frequency of my heterozygote. All right. So here my f of 1 is given as my 0.59 plus it will be half of my heterozygous, that is 0.16. So this is 0.59 plus half of 0.6 is how much? It is 0.08, right? So now if you add it up, you're coming to as 0.67. So option 3 is the correct answer. Option 3 is the correct answer. All right. So you get this type of question. So be very careful as to what they have given the data type. Accordingly, you select your own formula. Okay. So now, in a population, your genotypic frequency and your allele frequency will actually change over time, isn't it? Why will it change over time? Because in a population is subjected to mutation, it is subjected to natural selection, it is subjected to genetic drift, to non-random mating and gene flow. Now, mutation is something we all know, right? It is a change in the nucleotide. It can be a point mutation, it can be a chromosomal aberration. That also comes under your mutation. A change in the DNA structure, that is mutation. So, obviously, if there is a change in the DNA structure, the composition changes. Particularly if there is a base pair change, it will change the allele frequency. Okay? Similarly, natural selection. Natural selection, we know what? It is a biased selection based upon the fitness value. If you are fit with respect to the environment, the natural selection will favor you over others. So, accordingly, one genotype gets favored over the other genotype. So, that again shifts the entire equilibrium. It changes the uh, frequency of the genotype and the obviously the allele as well. Genetic drift. We will talk in details about the genetic drift in the later slides. Just for the time being, genetic drift is a random sampling. Okay, so it is a sampling error that happens, which again changes your allele frequency. Non-random mating. Another word for non-random mating can be selective breeding, okay, or inbreeding. Now, when you're doing selective breeding, that also changes your allele frequency, because again you are being very particular, you are being very biased. Whenever you become biased, obviously that particular genotype frequency increases over others. Okay. Now next is your gene flow. Gene flow can be in the terms of your migration. So when people are entering a population, they are bringing in new alleles, they are bringing in new variations. So automatically, see, you are counting the genetic variation or allelic variation over the entire population, isn't it? Number of the homozygous individuals divided by the total number of individuals in that population. So when your individual number increases or decreases because of migration, your frequency value will also change. Okay. Now, moving on to Hardy-Weinberg. Hardy-Weinberg stated that over the time, your allele frequency and genotypic frequency will remain constant. It will not change when all these disturbing elements are not present. So in absence of all these disturbing elements, why they are called as disturbing elements? Because they are bringing about a change in the frequency. So in absence of all these disturbing elements, all my genotypic frequency and my uh, allele frequency will remain constant in any population. That is Hardy-Weinberg equilibrium. So in order to summarize this Hardy-Weinberg model, we can take the help of this wheel here, which is telling you that first and foremost, Hardy-Weinberg assumes that in a given population, the population is large and not experiencing mutation, migration, natural selection, or sexual selection. All right? No disturbing elements. Next, the frequency of alleles and genotypes in a population's gene pool remain constant from generation to generation. So in absence of all these disturbing elements, the frequency will remain constant. Next, you have what? The frequency of the alleles in a population can be represented by P plus Q equals to 1, where my P is equals to my frequency of my dominant allele and Q is equals to the frequency of my recessive allele. All right. So in any population which is in par with the Hardy-Weinberg, my P plus Q will be equals to 1. All right. Now this is what is my allele frequency. What about my genotypic frequency? According to Hardy-Weinberg, my genotypic frequency is P square, just a minute please. Yeah. P square plus two PQ plus Q square equals to 1, where my P square is my frequency of the homozygote dominant, 2PQ is the frequency of my heterozygote, Q square is the frequency of my homozygote recessive. Now be very careful in the statements of Hardy-Weinberg questions what they are asking. If they are asking for homozygous, if they are saying that the number of homozygous dominant individuals is equals to say 10. When they are giving you a statement like this, the number of homozygous dominant individual is equals to 10, that means they are talking about p square is equals to 10. If they give you that the number of dominant individuals, number of individuals with dominant phenotype is equals to 20, that means they are talking about p square plus 2pq equals to 20. You know, these are the tricks. These are the things that you have to understand. You have to be very careful in the statements. Again, I'm repeating, homozygous dominant if they tell you, or if they tell you homozygous recessive, if they tell you homozygous heterozygous dominant, that means they are talking about a particular genotypic frequency. Okay, mainly this problem arises when we are talking about dominant. The confusion arises there. So it is p square equals to 10. But if I tell you that the number of individuals having dominant phenotype, okay, I haven't mentioned homozygous or heterozygous, I've just said dominant phenotype. That means it will include both your homozygous and heterozygous, because both are going to give you the dominant phenotype. If they tell you that the number of or the frequency of your the dominant allele is equals to 4, that means they are saying p is equals to 4. Don't write p square equals to 4, okay? They have clearly mentioned it is the allele, so it is p is equals to 4. All right, so these are the things that you have to keep in mind. So the frequency of the genotype in a population represented by p square plus 2pq plus q square equals to 1. So p square is obviously, as I've told you, it is the homozygous dominant. 2pq is the frequency of heterozygous. Q square is the frequency of the recessive. And lastly, the frequency of the allele can be estimated by calculating the frequency of the recessive genotypes. So if I tell you, it's simple. If I tell you that the frequency of the dominant allele is 4, okay? Then if I ask you what is the frequency of the recessive allele, it will be how much? 1 minus p is equals to q. So 1 minus 4 is equals to 3, right? Again, if I give you a statement like this, that the number of dominant individuals with dominant phenotype is say 20, okay, and the number of individuals with recessive phenotype is equals to say 16, okay, and I ask you to calculate the frequency of the dominant allele. That is, I'm asking you to calculate p. How will you be able to calculate? See, dominant phenotype means it is representing p square plus 2pq, right? So it is not possible from here to get hold of p. But when I'm saying recessive phenotype, that means this is my q square. From here, if I know my q square equals to 16, then I can find out my q, this square root of 16, that is 8. Sorry, not 8, 4. 4. 4 4s are 16, so q is my 4. Now, if I know q, simply 1 minus q will be my p. So 1 minus 4 is equals to 3. Got it? So this way we calculate. I hope it is clear to everyone. So in order to summarize, if you want to remember Hardy-Weinberg, what they are talking about, you can take help of the wheel. Okay, the concept wheel. Now, here is one question for you. Quickly try it out and let me know what is the answer. Okay. Bhakia Lakshmi, right. Okay, fine. I will do that. Okay, Vishal has already given me the answer. Okay, so every one of you are giving me the correct answer. Very good. Vishal, Kirtana, Anup. Yes, the correct answer is 42%. How are we getting it? Let us quickly solve this. It is given as the red hair is 9%. That means this is the recessive trait. So my Q square is equals to 9%, which is nothing but 9 by 900, which is equals to my 0.09. Okay. So from here I can find out my Q, the square root of 0.09, which is equals to 0.3. Now 1 minus Q is equals to my P. So 1 minus 0.3 will be equals to my 0.7. So I have got Q, I have got P. They are asking for heterozygous. That means they are asking for 2PQ. So 2 into 0.7 into 0.3. This is equals to your 0.42. If I place it in percentage, it will be 42%. Okay? Fine. So just a gentle reminder that if you wish to do more, practice more sums with us, practice more questions with us, go into the details of every topic, then you can definitely join us and we are here to help you out. So the Shaurya batch has already started and there are very limited seats left. Okay, so you can make your decisions quickly if you want. So just a summary of your Hardy-Weinberg equilibrium, coming to that. And after that, we will move forward to genetic drift. So Hardy-Weinberg, if they give you, start looking from here, okay? Don't, for the timing, don't look here. They have given you the number of individuals of a particular genotype. By the formula of genotypic frequency, you can find out the genotypic frequency, right? Actually, you can find out the allelic frequency, not genotypic frequency. Allelic frequency, and which formula to use here? The first one, 2 into the number of individuals who are homozygous A plus number of individuals who are heterozygous divided by 2 into the total. If you do this, 2 into 245 is 490 plus number of heterozygous is 210. So this value is coming as your 700 divided by 2 into the total. Total was your 500. So 2 into 500 is 1000. Okay. So your allele frequency P is coming as 0.7. Now again from here, similarly, you can find out your Q, which is 0.3. Now, if you find Q and if you can find out P and Q, you can definitely find out the probable genotypic frequencies. These are all the probable frequencies: P square, 2PQ, and Q square. Okay, so you can get this answer like this. This is just a summary. Okay, this is one type of calculation. This has got nothing to relate with the previous question which I showed you. This is another format that I have presented here. So now, another very important topic, which is your genetic drift. As I told you, genetic drift, this is a random sampling, right? Like one example, when you're walking, do you realize how many insects you have stamped? Or which particular insect you have stamped, which particular genotype you have stamped? Do you ever realize that? Or do you ever think and then put your foot on the ground? You don't, right? It is accidental. Accidental is a random sample. Okay. Now it might happen because of your stampage. A particular, insects of a particular genotype might die. So automatically your frequency, the genotypic frequency will change. Yes, that is one type of your genetic drift. Okay, that is one example of your genetic drift. That is one aspect. Another one is, suppose you have got an individual with a particular gene, okay, specific gene. Now, if you don't pass this gene, suppose this individual doesn't breed, okay, doesn't have any individual to breed with. So what will happen? These genes will not get passed on to the next generation. So whatever is the genetic frequency here, okay, say the yellow one and the red one. There's only one yellow and there are so many red. This will be maintained. But what if this yellow one breeds and passes on its genetic material to the progeny? Automatically, the yellow number will also increase. So now you will have a different genotypic frequency. All right. So whether you're passing the genes on or whether you're not passing the genes on, that will also determine your genetic frequency change, genotypic frequency change. All right. So this is your genetic drift, random sampling, which is also known as your Sever's right effect. Now, genetic drift, what are the features? Again, let us take help of this wheel to understand. Allele frequency of a population change over generation due to chance. Okay, so this chance factor is what is your genetic drift. Now, it occurs in all populations of non-infinite size. Actually, genetic drift is occurring everywhere, finite or infinite, whether the population is finite or infinite. Okay, but genetic drift has got a huge impact on a small population. All right, in a large population, the impact goes negligible. Next is effects are strongest in small population. Smaller the sample, the greater the chance of deviation from the predicted result. All right. The change, any kind of change, appears great when the quantity is small. When a quantity is large, no, any kind of change will not have that huge impact, will not get that importance. The same thing applied here also. Now, it does not take into account any alleles benefit. This is very, very important. If you compare it with natural selection, no, natural selection, as I told you, it is biased, is always favoring the alleles or the genes which are increasing your fitness. But genetic drift is a random sample. Okay, it doesn't pay attention to whether the alleles which are lost are beneficial or detrimental. So in course of time, you might lose out on important alleles, or you might have some harmful alleles getting fixed in a population. Okay, because it doesn't pay attention to what kind of allele it is. And two impacts of genetic drift are your bottleneck and the founder effect. See, suppose this is your source population. Okay, so as you can see, there are so many variations here. There is a blue, there is a red, there is yellow, there is green. All are representing different alleles, okay, of the same gene. Different alleles they are representing. Now you establish a new population. Now when you are, suppose individuals from here, from this source population are moving out. Okay, they're moving out to a different land, and they are occupying this land. They are now starting to inhabit this land, and they are increasing their population. Now which individuals are moving out is not regulated. Anyone can move out. Okay, so we might find that in this new land we have individuals comprising of the red allele, green allele, and yellow allele. There is no individuals of the blue. That might happen. Now, this is what is known as a founder effect. You have founded the population, your population, in a new place and with a new allele frequency. All right, this is founder effect. Now, it might happen that any population can undergo destruction by some reason. Might be a natural calamity. If you take the example of animals, then it can be hunting. Okay, any kind of a natural calamity. Because of that, that population can reduce in number. Now, take the example of hunting. When the hunter hunts down a tiger or any animal, does it pay attention as to the genotype of that tiger? What is the genetic composition and all? Does it sit and analyze that? No. He simply sights a tiger and just shoots it. So again, that is a random sample. Okay, so we might find that out of those tigers which are surviving, or out of those individuals who are surviving, maybe we have lost out on the greens. It can happen. So only the red and the yellow is surviving. So this shrinkage in the population size, this is what is known as a bottleneck. What is the effect of the bottleneck? Again, see, you have a new population with new allele frequency. Why? Because out of those who are surviving, only they are reproducing to increase the population. Okay, so this is again a new population after the expansion and with new allele frequency. Now, in this picture, they have shown all the things together in one. Okay, the founder effect, the bottleneck effect. It's not necessary that founder effect and bottleneck effect is happening in the same population. Understood? It's not necessary. Again, it can happen. So this is just one diagram to explain all these effects of genetic drift. So again, let us take the help of this concept table to understand the difference between the founder effect and the bottleneck. So founder effect is what? The phenomena which occurs when a small group of individuals become isolated from a large population, and bottleneck effect is the phenomena which occurs when a population rapidly decreases in size. These are the key words. These are the keywords. There is a decrease in size for bottleneck. From here is what? They become isolated from a large population. These are the keywords. So in a statement, when they give you, you have to look for the keywords to understand. So founder effect can be one origin for the bottleneck effect. Bottleneck effect can have different origins. All right. As I told you, any kind of natural calamity. So migration of very small individuals to the main from the main population, this can be one of the cause of founder effect. Whereas there's a sharp reduction in the population because of environmental events, that is your bottleneck effect cause. Founder effect does not affect the original population, but bottleneck definitely affects the original population. Okay, the entire composition of that original population changes. Founder effect produces a population with a non-random sample of genes, whereas bottleneck occurs due to random sampling of genes from the original population. Right? So, I hope it is clear, everyone. So, let's move on to the last section, which is the deviation from null hypothesis. So, we have already seen that Hardy-Weinberg is stating that there will be no change in the allele frequency or genotypic frequency in the population in absence of disturbing influences, right? Now, that is an idealized condition, a very idealized condition, because obviously, who can stop natural selection? Who can stop genetic drift? You can't stop it. Okay? But still, Hardy-Weinberg is very important for any kind of population study. Why is that? Because it tells you how much you have deviated from the standard. Hardy-Weinberg is your standard. Okay, and when you deviate from that Hardy-Weinberg, or when you deviate from that null hypothesis, it tells you how much you have deviated, and that leads to further studies as to what is the reason for that deviation, whether it is mutation, whether it is migration. Now if there is migration, then what will be the future of that population? So many questions comes one after the other once you have made your the deviation, once you have understood that yes, your population has deviated. So first and foremost, mutation is one reason for deviation. We have already seen. Okay. Now, because of mutation, the new allele frequency will be, which is depicted as Q prime. Okay, I'm talking in respect to the recessive allele. This is given as, it is calculated as mu by mu plus nu, where mu is equals to the forward mutation rate, and nu is your reverse mutation rate. All right. Similarly, if I write in the terms of P prime, okay, P dash, which is my new allele frequency after mutation, it will be written as nu by mu plus nu. All right. Now, in this limited time, obviously, I don't have that much liberty to explain exactly why we are getting this formula. Maybe if anyone joins our regular course, or if there is anyone from the regular course right now, for you it will be clear, because we discuss all these things in depth there. So for the timing, I'm just giving you the very important points. Okay, you can elaborate on these points. So that is how you calculate the new allele frequency after mutation. Now, non-random mating is another disturbing element. Non-random mating can be in form of inbreeding. Okay, it can be in form of inbreeding. Now, inbreeding, the magnitude of inbreeding is calculated as F. It is depicted as F, and it is calculated as F equals to 1 minus observed heterozygosity divided by expected heterozygosity. Okay, so this is equals to 1 minus observed heterozygosity, what you are calculating, divided by expected is 2pq. Fine. Next disturbing element is your natural selection. Now, natural selection is based upon two things. There are two parameters here. One is the fitness level, how much fit you are, which is depicted as W. And then there is selection coefficient, which is given as 1 minus W. All right. Now, selection coefficient is simply determining how much you are against a particular genotype. All right. So, if your selection coefficient is greater is high for a particular genotype, means that genotype is unfavorable. Okay. So, how to calculate my new allele frequency after natural selection? If I write the genotypic frequency, then my new genotypic frequency will be written as p square into the fitness value of that particular genotype, which I'm writing as WAA, divided by the mean fitness of the population. All right. For my heterozygote, it will be 2pq into the fitness of the heterozygote, fitness value of the heterozygous, divided by the mean fitness of the entire population. For the homozygous recessive, it is going to be q square into the fitness value of that genotype divided by the mean fitness value of the entire population. All right. So if you know the genotypic frequency, from here you can definitely find out your allele frequency also by the same formula what we have discussed at the start of this presentation. Okay. So that goes with your natural selection. The next disturbing element is your migration. Now, migration, the change, the new allele frequency after migration is written as Q2. Okay, I'm writing as Q2. This is representing the recessive allele, the frequency of the recessive allele in this in population 2, which is facing the migration. That means you have a population which is population number 2, where people are entering. There is a lot of immigration here. Then the allele frequency change in this population is written as Q1 into M plus Q2 into 1 plus M. Okay. Now, Q1 is what? It is the frequency of the recessive allele in the population 1 from where the people are migrating out into the number of migrants or the immigrants. All right. How many people have moved out? That is your M into their frequency in their original population. Plus Q2 is the frequency of the recessive allele in population 2 into the number of natives, okay, which is 1. sorry, 1 plus m, no, this is 1 minus m. Okay, into the natives is your 1 minus m. Fine. That is your migration. Now, if you know q, you can find out p. So p will be simply 1 minus q2. And lastly, you have genetic drift, another disturbing element. So genetic drift, how do you calculate the magnitude of genetic drift? It is written as sp square is equals to pq by 2ne, where ne, this is known as the effective population, effective population size. See, in a population, not everyone are fertile. Not everyone is reproducing. Okay, and in a population, only those who are reproducing, they are under the calculation. Why? Because they are the ones who is adding individuals to the population. Okay, so in order to find out how many individuals are capable of reproducing, we use this value as NE. We use this parameter NE, the effective population size. All right. So your Hardy-Weinberg equilibrium, it deviates from the null hypothesis, from the standard, because of all these effects, and I've already shown you how they are deviating. So with this, I come to the end of my presentation. You can practice some sums from here also later on. All right. So I hope it is clear to everyone. You do get questions from here. Like, let me quickly just go through this question. Like they have told you, in each generation, A1 mutates to A2 at the rate of mu equals to this, okay? While A2 mutates to A1 at the rate of 2 into 10 to the power of minus 5. So assume that the population is infinitely large and no other evolutionary force is acting. The equilibrium frequency of A1 is how much? So you just have to use the formula P is equals to nu by mu plus mu in order to get the answer. Okay. Another type of question, if you see, this is based around your inbreeding coefficient. They have given you the frequency of the two alleles. They have given you the frequency of the heterozygotes. And they are now asking you to find the inbreeding coefficient, which is F. So again, you know the formula. F is equals to 1 minus H by 2PQ, right? So H is given to you, which is 0.28. 2PQ is given to you, 2 into 0.3 into 0.7. So you just have to solve it to get to the answer. See, these are so direct questions you get from this part. Okay, you just have to have a very clear concept as to why it is happening, how it is happening, in order to remember. So I hope you have now got some concepts about how to approach with this topic. Let me hear some reviews. Did you understand? Did you find it helpful now? Okay. Okay, so I'm really so glad that you have found it helpful. Thank you everyone for your attention and wish you all the best. Prepare it in this way, prepare the chapter in this way, and definitely practice the questions. Okay, you will definitely be able to solve all the answers from here. So wish you all the best. Thank you, and I will take a leave now. Bye-bye.